Hello, good afternoon, everyone. We are back for another exciting lecture, and this time is about protein synthesis. Okay, it's a lot more manageable in terms of the length. It's only 55 slides, but we do have quite a bit of problem solving to do in this particular lecture. Okay, so I hope you print out the notes and you have it in front of you. Yeah, let's get to it. So we learned about DNA replication and how the cell divides in lecture 6. And now we're just gonna, take that a step further, and we want to try to understand the relationship between the DNA and the protein. Specifically, you know by now that the DNA contains the instruction to make the protein, right? The AC and the T, ACTs and Gs, they actually are instruction to make the protein. But exactly how does that happen? Right? So we're gonna learn how to decode the genetic information and actually build a protein, um, together. So we're going to start with the transcription process. Now, if you've forgotten about these words, let me just give you a quick reminder. DNA, it's going to get copied into RNA, and that process is called transcription. And then from RNA, we're going to use the information to make the protein. Does anybody remember what that's called? DNA to RNA is transcription, but from RNA to protein, what is that called?
Translation.
Excellent. Okay, translation. And so that's what we'll be learning for the whole lecture, right? Probably not going to finish today. I am pretty optimistic that we will be able to finish, this is the wrong class, that we'll be able to finish this on Monday, okay? So definitely come to class on Monday. But I'll tell you about the quiz. Like I don't know where to put it yet. I just need some more time to think about it. And of course, there is also the lab that's going to be happening next week. Okay, some terminologies to help you, uh, understand all the terms. First of all, the genome. The genome of an organism, it's all of its genetic material. So think of the genome as the whole textbook, okay, the entire textbook. A gene is a section on the chromosomes that contains instruction to make the protein. So the chapter here is like the chromosome. Okay. So your entire textbook has 46 chapters. Right? 46 chapters. Right? 46 chromosomes, right? That's how many we have. And the whole textbook is made of letters, right? In the textbook, the alphabet is A to Z, but in DNA, the alphabets are A, C, T, and G. A gene is going to be a section of the chromosome. So like within the chapter, you have chapter 1.1. 1.1 is going to teach you about something. 1.4 is going to teach you about something. You also have, in another chapter, like chapter 4, 4.1, 4.2, 4.3, right? Those sections, it's like a gene. Right? And each section gives you information about a specific topic. Now, where do we find the gene? The location of the gene on the chromosome is called the locus. And the locus is like the page number. Page 52, that's section 1.5. Okay, so we use a similar numbering system to locate the gene. I'll tell you more about locating a gene and why that is important when we get to the biotechnology lecture. Okay, but locating a gene is important if you want to clone it. Right? All this information is freely available online. If you want to look up a specific gene, you can actually search it on the internet. It will tell you exactly where it is on a particular chromosome. So any question about genome, gene, chromosome and locus. I hope the analogy helps a little bit here, okay? So previously, I told you about our chromosomes is 23 pairs. Half of it is from our mother, half of it is from our dad, right? And the two chromosomes, even though they are similar to each other, but they're not identical. The copy, let's say chromosome 5. We will take chromosome 5 as an example. The chromosome 5 that we inherited from mom is not identical to the chromosome 5 that we inherit from dad. They are similar, they are the same size, they are the same length, and they actually contain genetic information in the same order. Okay, so for example, if there are 4 genes, in reality there are hundreds of genes, right? But if there are 4 genes only, and the order goes A, B, C, D on one, it's going to be the same order on the other strand. Okay? A more concrete example, if you have a gene here that gives you eye color, you will find the exact same you'll find an eye color gene at the exact same location on the other chromosome. Now, are they necessarily the same eye color? Well, it depends. If both your parents have brown eyes, then it's possible that you will have the same gene. So we represent gene using capital letters and small letters. In this case, it's like big A, same gene. More likely than not, your parents are gonna have same eye color, right? Generally speaking, people tend to measure others with the same ethnicity, right? I mean, sure, you can marry other people as well, but generally, I'm saying, right? So like both parents probably have the same eye color, both brown eyes, both blue eyes, what have you, right? So the gene for the eye color might be the same. But when it comes to other things, it might not be the same. Let's say, I'm just totally making this up, okay? Let's say this location is for blood type. Right, then you will find blood type at the same locus on the other chromosome. And, your parents might have type B blood. The mom might have type B blood, but then the dad might have like type O or something. Right? So the gene, even though they encode for the same type of information, blood type, but it might not be the same version. Okay, so like if you go down all the way, the information that it encodes, it's for the same trait in the same order, but the version of it might be a little bit different. So different version of a gene is called an allele. Allele. So brown eyes, blue eyes, that's an example of different allele. Type A blood, type B blood, different alleles. Okay? And then the chromosome here, these chromosomes, we call them homologous chromosome right here, homologous chromosome. So they are same size, same order of information, but the alleles, the version on them might be different. Okay? So chromosome 1 and 1, they would be homologous. 2 would be homologous to 2. 3 would be homologous to 3. So on and so forth. Okay? 4 is not homologous to 5. 4 is only homologous to 4. A way to think about this is like if you think about you have like 23 pairs of shoes, okay, and you randomly scatter them on the floor. You would have no problem matching up the left shoe and the right shoe, right? Because they look similar, right? But the left shoe and the right shoe are not identical, right? You cannot fit your right foot into the left shoe, right? So it's kind of like that. The homologous chromosomes are very similar, okay? But there are slight differences. Questions? Okay. So this is the central dogma of cell biology. You start with the DNA, and I already told you through the process of transcription, you're gonna make a pre-mRNA. This is new to you, okay? A pre-mRNA is like, it's not ready yet. We gotta fix it up, and to fix it up, we do what's called transcriptional modifications, okay, which is same as RNA processing, the same thing. Same. We need to process it. We need to improve its stability so it's not going to break down too quickly. And then we will get the actual RNA. And that's going to leave the nucleus and go into the cytoplasm. So translation happens in the cytoplasm, right? Whereas these transcription and RNA processing happens in the nucleus. And the translation is where the ribosome will use the information on the RNA to make the proteins. And I'll show you how that is done today. All right, just stop me if you have questions. Okay, I'm not gonna keep on asking if you guys are okay. I'm sure you are a little bit annoyed by that. Purpose of transcription. Why do we have to do transcription? Why not just, take the information from the DNA directly and make a protein? Well, it turns out, we talked about this before as well. The DNA is too big to leave the nucleus. Right? The nucleus has only little pores. It's not possible to take the DNA out. That's why during mitosis, you actually have to dissolve the nuclear envelope, right, for the DNA to come out. And we don't want the DNA to come out necessarily. Right? They might break down and stuff if they're in the cytoplasm. And so to prevent this problem, you have to copy only the portion of the DNA that you need. Right? I forgot who it was that asked this question last time. I think it was Fatima. Is it like, we have all the cells in the body have the same set of DNA, all the same set of chromosomes. But like, which one do we turn on is going to depend on the kind of cells that you have, right? So when I say turn on a gene, it means you're going to be copying the gene. Okay? You have all these recipes in the cell to make all the different proteins, but you don't make all of them all the time in every cell. Okay? You only make the ones that are appropriate for that particular cell. Okay, so you would turn on the gene, you would take the recipe out, make a copy, and then use it to make the protein. So this also adds a level of control, right, to see when to turn things on and when to turn it off. Even if you are supposed to make that protein in a particular cell, you are not continuously making it necessarily. Sometimes you only need the protein in certain situations. Then you only need to turn on the gene when the occasion arrives, right? Okay. Same thing. Like think of like cooking recipe, right? Like so many recipe. You're not gonna make chicken every day, okay? Or like you're not gonna make turkey every day. You're gonna make turkey on special occasions, right? Or whatever it is that you make. So recall base pairing rule. A goes with T, G goes with C. You know that already. That's between DNA and DNA, right? But it turns out DNA can also pair with RNA with one key exception. Where there is thymine in the DNA, we are replacing it with uracil. Now, this is not an entirely new concept to you. When we learned about the helicase and the primase and all this stuff, remember when we put the primer on, we are following A pairing with U. Okay, so we know DNA and RNA can pair together under certain conditions. Okay, so transcription, DNA to RNA. It happens in the nucleus of the cell. We know that already. And to make this happen, the enzyme that we need is RNA polymerase. To build DNA, we needed DNA polymerase, right? Now we are making RNA, so of course we need RNA polymerase. Okay, thank goodness there's just one. Okay, there's no 1, 2, 3 or anything like that. Okay? It's just RNA polymerase. So this one, it kind of does everything. Okay, it unwinds the DNA, it copies the DNA and make it into RNA, and then it's gonna close the strand afterwards as well. Right? Unlike DNA replication, where we unzip it and we never put it back together. This is different. And remember, you have the whole DNA. You only open up the parts that you want to copy. After you copy it, you gotta wind it back up. Okay, as if you never touched it. Right? You need to leave it as it was. So the RNA polymerase will open it, copy, and then zip it as well. Okay. So the product is called the pre-mRNA, which actually needs further processing before it can leave the nucleus. And I'll tell you what those modifications are in a bit. But first, we have to take a closer look at how the transcription process takes place. Okay. So here it's just on the top right corner, it's just trying to refresh your memory a little bit about the relationship between the adenine and the thymine. Right? Adenine bonds with thymine with two hydrogen bonds. Okay, with two hydrogen bonds, and instead of three, right, between C's and G's. So it's easier to pull apart the DNA when there are more A's and T's, and that's what's happening here. You have a let me just draw this out for you. There we go. What is this? This is March 13. All right. So, this is DNA. DNA, double helix. Okay, and then I'm just gonna draw it in a rectangle. It's easier that way for you to visualize. Okay, but know that it's it's still continue to be double helix in between. Okay, so like that. Now, so this whole thing would be a gene. It's a segment of the DNA, right? But how do you know it's the beginning of a gene? It's just a bunch of A, C, T's, and G's, right? Well, similar to like a textbook. How do you know it's the beginning of a section? Because it will tell you, right? Like 1.2, and then it will give you the title, and then usually it might have like a picture that relates to the topic. So similarly, in the gene, there is like a special sequence at the front, and that is called the promoter. Promoter. It's AT rich, and it marks the beginning of a gene. It's AT rich because it's easier to pull apart, only 2 bonds instead of 3 bonds. So the RNA polymerase is going to be able to find and bind to this promoter. Okay, and it's going to move along and open up the DNA. Right? In fact, as the polymerase moves along, it's going to unzip the DNA at the front and rezip the DNA at the back. Okay, so you only open a little bit of it at a time. And at the same time, it's going to be copying the entire strand. Now, when do you stop? So again, in the textbook, you would know it's the end of a section because there might be some, review questions. Yeah, there might be some summary bullet points. But in the case of a gene, the end is marked by what we call a terminator sequence. Terminator sequence or terminator. It marks the end of a gene. Okay. So here,
Professor,
yeah,
there is just one gene in a DNA.
no, thousands, hundreds of genes, okay? Just like in a chapter, you have 1.2, 1.3, 1.5, like so many sections, right?
Okay.
Yeah, so in reality, you know, you might have a gene here, you might have a gene here, you might have a gene here, and then you would have some DNA in between that doesn't code for anything. Okay? It's not really showing up. All right.
I have a question, Professor.
Sure, go ahead, please.
So in the gene, the terminator terminates the growth of the gene?
No, the terminator is going to be the end of a gene. So if you want to copy this gene, when you get to the terminator, you need to stop copying because otherwise you would copy too much, right?
Oh, okay.
Yeah. Let me see if I have a picture here to show you guys. Give me a moment. Yeah, so typically this is what it looks like. Like this is just a random chromosome that I search up. Okay, and on this chromosome, right? Like that's a gene, that's a gene, 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 gene. Some genes are longer, some genes are shorter. Okay, but like this is not even accurate representation necessarily because there's like hundreds of genes that are on a chromosome. Okay, so over here we have one gene. Chromosome is really long. Okay. Any other questions? These are great questions, by the way. Anything else you want me to clarify? Just remember, if you have a question, someone else probably have the same question. Okay, so to start the process of transcription, RNA polymerase binds to a region on the gene called the promoter. And this marks the beginning of a gene. The promoters are rich in adenine and thymine, making it easier to be unzipped. And the RNA polymerase is going to unwind the DNA strand and start RNA synthesis. And the kind of RNA that we are making is called mRNA. The M stands for messenger. Messenger RNA. Remember, there are three types of RNA, right? There's messenger RNA, transfer RNA, as well as ribosomal RNA. So the one we're making right now is messenger RNA. Okay. So next is elongation. Initiation, elongation. The RNA polymerase moves along the DNA strand. It will unwind the DNA ahead, copying the current section, and then rewound the DNA behind. And of course, they have to use the base pairing rule, right? The DNA and the RNA base pairing rule. Now, this is where things gets a little bit confusing, perhaps. DNA is double-stranded. There are two strands here. So you can see the top one is light blue and the bottom is darker blue. When you build the RNA, you are only building a single strand, right? RNA is a single strand molecule. So you will only be copying from one of the two DNA strands. Right? I will explain this a little bit more in the slides to come. But the one that we are copying from, that's called the template DNA. And we're going to be following the base pairing rule. Okay? T pairs with A pairs with U, because we're building RNA, C pairs with G, and so on and so forth. So the template DNA is the one that the RNA polymerase is building the strands from. The other strand that we are not using, that's called a coding strand. I'll tell you more about them later, but now I need to know if you understand why we are not copying both strands and that we are only using one strand to build the RNA. Right? So again, the ones that we are base pairing with, that's the template DNA. And the one that we are not using, it's called the coding strand. So this continues. Okay, you're going to be rezipping the stuff at the back, and then you're going to be unwounding the stuff ahead. And this big grayish looking thing, that is the RNA polymerase. So this is elongation. It's going to continue all the way until when? Someone please answer me. When are we going to stop this copying? Until we get to the
termination part.
That's right, the terminator. Okay. So RNA polymerase will eventually reach a region on the gene called the terminator sequence. The pre-mRNA transcript is released. Right, that's the purple thing right here. That's the that's the pre-mRNA. That's the copy that we just made. And then we no longer need to copy more. So the RNA polymerase is going to fall off. Right? The terminator sequence is where we're going to stop this transcription process. Okay. Now let's try to understand more about template strand and coding strand. So the template strand is the strand that is used as a template to make mRNA copy. The way that we build the way we build nucleic acids in general, whether we're building DNA or RNA, is that we have to use base pairing rule. Okay? Like if you have A, C, T, T, G, C, something like that, if you need to build something from it, your body only knows how to build a complementary strand. It's not gonna know how to copy it directly. If you tell the body to copy this strand, it will not be able to do it like this. Okay, it can't do a direct copy. Because of the way that the polymerase works, they can only build the complementary strand. So like, U, G, A, C, G, that's what it will do. Now, that's gonna be a problem because if you copy it using the base pairing rule, you end up having not the exact same thing, but you have the complementary strand, right? You end up making a copy of the other strand that you didn't use. Okay, let's go back to here and see. When we build off the template strand at the bottom here, we ended up having a strand that's the same as the other one. Look, the C matches with the C, the A and the A. T and U essentially is the same thing. C, 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 A. Right? You understand? You end up having a copy of the other strand that we did not use. So it's like minting a coin. This is a mole for minting a coin. The mole has to be flipped compared to what you actually want to make, right? If the mole is the same as what you want to make, then it's going to come out backwards. Do you understand that? Right? The mole has to be opposite to what you want to make. And that's kind of like the situation here. The two strands of DNA, the coding strand is the one that actually contains the code, that actually contains the information on how to make the protein. So that's the one we actually want to have a copy of. But we cannot copy directly from the coding strand because it will come out backwards, which is why we're going to have to use the template strand. The template strand is like the mole. And when it comes out, it will be looking exactly like what we wanted. It will come out looking like the coding strand. Okay, I'm just going to pause for a second and see if you guys have any questions. It's like a little bit difficult to understand that concept. Any questions you want to ask so I can clarify it for you? Really? Crystal clear? Yeah, go ahead.
Yeah, the template is like a sample.
The template is I wouldn't say it's a sample, but it's what you will follow
Yes,
to
yeah.
build. Yeah.
Okay, okay.
Yeah. And again, it's because of a limitation of how we build things, right? We can only build using the base pairing rule. Your body does not know how to copy directly. All right. So we're gonna have to know how to do things like these. Right. Here it says, given the template strand, find the coding strand and the mRNA. Right, so this is template. Template. Again, just want to reiterate.
Vincent,
Yeah,
lost the previous slide.
I am
Please.
sorry.
The previous slide, please. I want to.
Yeah.
So for the template strand, so the mRNA, the mRNA is one making the DNA copy, right?
The mRNA is a copy of the DNA.
Of the
Yes.
DNA, right? Yes.
Mhm.
So when we I don't quite get the like the coding using the backward. I understand this when it comes to like printing money and things like that. But then why do we have like where do we have a template when like we already have something we can just copy directly like that?
Yeah, I understand. Okay. So look, much like making a coin,
Yeah.
we cannot copy directly. It's different from like photocopying, let's say, right? Photocopy, you put in a photocopy machine and it comes out exactly the same as the original, right? When it
Okay.
comes to minting the coin, if the mold looks exactly like the actual coin, do you understand that it will come out backwards?
Yes, I
Okay,
do.
that's right. So when we make when we make RNA, when we make a copy of the DNA in the form of RNA,
Mhm.
this is like the coin. It's not like the photocopying.
Oh,
Meaning,
okay.
meaning if you give RNA polymerase, like if they see an A, they will not be able to build an A because that's like photocopying, right?
Mhm.
When the RNA polymerase see an A, what is it gonna build instead?
The backward side of A,
Which is, what does A pair with?
T.
That's right, but we're building RNA, so there's no T. Instead we
Okay,
have
you're asking.
exactly, right? So like you see an A, it will build a U. It sees a C, it will build a G.
A G, yeah.
That's right.
Okay.
So
Got
you
it.
see when it uses the template strand and then you build according to the base pairing rule, what you end up making is not the template strand. What you end up making is the coding strand,
Coding
which
strand,
actually
right.
contains the code, right?
Okay.
Yeah.
Thank you.
So very good. But again, if we use the coding strand and build it instead, then we would end up losing the code and end up having the template strand. Right? It's exactly same as making a coin with the wrong mold and the money is gonna come out backwards, which is useless. Okay. I hope that's a little bit better.
Excuse me, Professor.
Yes, go ahead.
I don't understand the difference between messenger RNA and RNA.
Okay, so RNA are different kinds of RNA, right? A long time ago, we said there is messenger RNA, there's rRNA, which is ribosomal, and then there's something called tRNA. So they all have different jobs. For example, you already know the rRNA makes up the ribosome, right? Do you remember that?
Yes, Professor.
Yeah. And now we have something called messenger RNA. The mRNA contains a copy of the DNA. That's all it means. It's a messenger. It relays the message in the DNA to make the protein. That's basically what it means, right?
Okay.
Yeah?
Okay, good.
Okay.
Thank you.
All right. So let's try this example together and hopefully it'll make it a little bit clearer. We have the template here. Now, if you have this strand here on the screen, am I going to be able to make the protein from it? Does this contain the instruction to make the protein? Sorry, what did you say, Mary? I cut you off. I'm sorry.
No.
No, this is just a template. This is like the mold of the coin. It's backwards, it's useless. It doesn't have any value to it, right? Okay. So what would the coding strand be? Well, coding strand is just the other side of the DNA, right? So you can write out the coding strand. What's the first one? Help me out, people.
T.
That's right. And T is good because this is DNA, right? Remember, both of these things are on the DNA. So this should be easy. All right. That's a coding. All right. So now we want to make the RNA. Okay, the RNA. Well, the RNA, if we actually go through the process of building the RNA, it's going to use the template strand, right? It's going to use the template strand and it's going to follow the base pairing rule. So what does A pair with on the RNA?
U.
U, that's right. So there is the U. And the next one is also a U. And then G. T pairs with A. So on and so forth. So in the end, the RNA, the messenger RNA, is the same as the coding. Same as coding. But T becomes U, right? Just because we're doing RNA and there is no thymine on RNA, that's all. Okay, so that's why using the template, it's gonna give you the coding. And that's what we want, because that's where the codes are. That's where the instructions are. Okay. Can you give me one word to describe the relationship between the template strand and the RNA? One word. What's the word I'm looking for?
Transcription.
Okay, yeah, that's the process to make it, yeah. But in terms of the relationship, how is the template strand related to the mRNA? One word that starts with C.
Complementary.
That's right, complementary. Right? They follow base pairing rule. It's just that, you know, A goes with U instead of T. All right. Let's try another one. Create an mRNA from the template strand here. Okay, you try. Okay. The template strand. Now you don't actually have to write the coding strand like we did. Okay, template. You can actually go directly to the mRNA because we just found out that the template strand and the RNA strands are complementary. You can go ahead and build that directly yourself. I'm going to give you a moment to do that. You can type the answer in the chat box. Actually, I would appreciate it if you're able to type it in the chat box so I can see. Okay, Aminat, thank you. That is wonderful. Anyone else? Okay. So you just follow the base pairing rule. A pairs with U, T pairs with A, and so on and so forth. You have to ask me questions if you don't understand. It's only going to get more confusing as I give you more information. So please, anything that I can do to make this easier? Okay,
Professor, excuse me.
sure.
Where is the location of template is on DNA?
Where is it? Is that your question? Yeah, so
Yes.
we don't have to know that. That is a difficult question to answer. But it's a good question. Okay, so in this picture, it looks like the template strand is at the bottom, right? And then the coding strand is on the top. But that's just a drawing. We don't know. If I give you two strands of DNA without more information, you wouldn't be able to tell whether it's coding or template. So I will always have to tell you that information in the question. Okay, I will always have to tell you whether you're looking at a template or coding. All right. So for example, if I just give you this strand, you won't know if it's coding or template. And that's why in the question it says create an mRNA from the given coding strand. So I told you that this is coding strand in the question, and you're like, oh, okay, this is coding. So please try to make the messenger RNA from the coding here, please. Mhm. Anyone else? That's okay, Fatima. You spot the error, maybe? Anyone else? Please, I need to know if you get this, guys. Okay, thank you. Thank you. Okay, so far no one has it right, but I do appreciate all of you trying. Anyone else? Okay. Let's go back to here. Now you have the template and you have the coding, right? What's the relationship between template and mRNA? Complementary. What about the relationship between coding and mRNA? Look at the coding strand and the mRNA. Look at them. What's the relationship? What's the relationship? Look at them. Look at them. What's the relationship?
Oh, okay. So the guanine and the A's and the C's are the same except for the thymine and uracil.
Yeah, so basically they're identical, right? Isn't it?
Yeah.
But T becomes U.
Yeah.
Because there is no thymine on
Oh,
RNA, right?
okay.
Okay.
Right.
So when you have a coding strand on the test, you should be happy because so little work. You just copy it. Okay. But of course the T has to become U. And then everything you just copy. Because I told you the coding strand is the instruction. That's the one that you want to make a copy of. That's the one that you need the mRNA to be identical to. Okay. But in the cell, to make this happen, you still need to use the template strand, right? But for the purpose of writing it out, like we can skip writing that part out because we know the messenger RNA is same as the coding strand. Okay, I think I've lost some people there between example 2 and 3. Example 2 is the template strand. That's why we have to do the base pairing rule. If it's a coding strand, we can just copy directly. But of course, changing the T to the U. I'll wait a little bit, okay? For you to let this sink in and, ask stuff. It's very important you ask.
Professor, if we, for example, if you ask coding from coding crate to DNA, how can we
But coding is already DNA though, right? Is it?
DNA? Okay, okay, yeah.
So, maybe you're asking about this. Like, what if we want to figure out the template?
Okay, yeah.
Yeah, what would the template be?
That's from coding.
Oh yeah, from the coding. So what do you think the first one is gonna be? You want to try?
I think I have to think first.
Sure. Or anyone else is okay. The code, we have a T here. What would it be on the other side of the DNA?
Basically, if it is a template, we don't have the uracil.
So what's the T pair with in the DNA?
I mean with adenine.
That's right.
And
So it's gonna be A.
yeah,
And then the A is gonna pair with the T,
with the
right?
and G with C.
That's right.
Yes.
And so on and so forth, right?
Yes.
So in reality, the polymerase is gonna use a template to make the mRNA, but it doesn't change the end result. The end result is that the mRNA is gonna be identical to the coding, right? Okay.
Before you go, I really want to clarify something. I think I'm mixing
Sure.
it up. So RNA is making a copy of DNA.
Yes, but
Okay.
only one of the two strands of a DNA, right?
Oh, okay.
Yeah,
Okay.
we're not copying both. We are only copying the coding because the coding is the instruction, right? But in order to make the coding, we have to use the template to make it. Agree?
Yes, thank you.
Okay.
Are we gonna do this in the quiz, like make an mRNA?
Yeah, you bet you will. That's right. I'll show you a question, okay? You don't have to type it out, okay? Thank goodness, right? You just have to choose the right answer. But to summarize, okay, this is it. Okay. Now, I hope I done a reasonably okay job explaining this to you. But after, if you think about this a couple more times and it still doesn't make sense and you're like still super stuck, this is the cheat sheet, okay? You're gonna put this on a sticky note and then stick it on your monitor in case you get confused on the test. All right, so let's read it together. Coding strands and mRNA are identical. They are the same. They have the same information. It's just that uracil replace all the thymine. So if I give you a coding strand, basically change all the T's to U's, which is what we did here. We change all the T's to U, everything else stays the same. If I give you the template strand, you have to understand the template strand and mRNA are complementary. So you use the base pairing rule. A pairs with U, T pairs with A, and C, G's, so on and so forth, right? Those are basically the only rules up to this point. Is it going to get more confusing? Yes, unfortunately, but we will get there soon. Yeah, go ahead,
Professor,
Lika. Yeah.
the template strand are not complementary with coding, just
Yeah.
with
Oh, yes.
mRNA.
Template and coding is also complementary. Yes.
Okay.
Okay. But here that this slide focuses on the relationship between the DNA and the RNA, right? Okay. Okay. Maybe one more. Okay, like cheat sheet. Okay. Template. Okay, really, there are only 4 letters, right? A, T, C, G. All right. Tell me please, what is on the coding? If it's A, T, C, G on the template, what is it on the coding?
A with T,
Pardon me, sorry, one more time. I didn't quite catch all of it.
A, T, T is T, A, I said
Yeah,
T,
yeah, okay, T, yeah, T. Sorry, yeah,
yeah,
T, yeah,
A,
T.
A, and then G,
Uh-huh.
and then C.
Okay. Now, do we all agree? Do you guys have something different? Okay. Well, this is correct. These are DNA, right? Complementary. Okay, what about the mRNA? What would the mRNA be?
U,
Okay.
and then T, no T, yeah.
UA,
U,
Mm-hmm.
GC.
and Gen
GC.
C.
That's
Yeah.
right. Okay. That's all. Okay, so maybe put that on a sticky tag instead, right? That's all. Like really, that's all. Because there are only 4 letters, right? And they just use the same rule for the rest of it. Are we okay? Let me know, please. What can I do? It'll get better. Okay. It will
Okay,
get
so in the test, we'll be told if it's DNA or mRNA, right?
on the test. It'll be like this. Okay. So let me start the poll. You guys tell me. Now, I would suggest when you see a question like this on the test, don't look at the answers. Right? It's going to confuse you. Just work it out yourself first, and then try to match the answer to your match your answer to the choices. Okay. Okay, I only have 4 answers. I need more, please. Anyone else? Okay, well, thank you for those of you who have participated. Let's take a look at the answer here. We are given the template strand right here, template strand, and we're trying to build the mRNA, right? So if the template is given, then the mRNA is going to be complementary. So complementary to TTA is AAU. Okay? And then you might not have to do the rest because you just have to check, right? Like that's not AAU, that's not AAU, that's not AAU. So the correct answer is C. I mean you can do the rest just to be sure, but the answer is C. Okay, for those of you who chose A, that would only be the answer if this was a coding string. Coding, you don't change them, right? Okay, different version of a gene is called, I'm just gonna tell you this one, okay? You can check it out later. We're running out of time. Allele. Okay, blue eyes, brown eyes, different alleles. All right, now we're gonna take a note on post-transcriptional modifications. Okay, so here, let's take a note. So, from the DNA, we copy an RNA. We copy an RNA strand, and so this is RNA strand. But on the RNA strands, it turns out there are some regions that do not code for anything. Okay, it turns out that you copy some like useless information, so to speak. They are called filler DNA, junk DNA. So RNA also has direction to it, 5' to 3' like that. And so we have these coding sections. Okay, these are called exons. Those are the coding sections. And we also have non-coding sections. These are introns. Non-coding sections. It's kind of like this. Like if you were gonna copy like an article from a magazine, let's just use that example. And you want to copy an article in the magazine for your research project, right? And then when you copy it, there might be some advertisement page in the middle of an article, right? Well, the advertisement is not useful for your purpose, so you're not gonna copy it. Right? Even after you copy, you're gonna rip it out. You take it away. You don't need it. Right? That's what the introns are. The introns are non-coding sections. Okay, they don't actually contribute to making the protein. They are filler DNA, junk DNA. Why are they there? Well, we don't have to worry about it. It's like I already give you too much information. I'll tell you about everything. This is no longer an introductory course. Okay, they're there for a purpose, but for now we don't need it. So in post transcriptional modifications, first thing you need to do is to remove introns and connect exons. So it's like, you know, an exercise of cut and paste. You chop them out, you literally chop them out, and then you end up having only exons, right? Okay, so like exon, oops, oh my goodness. Exon 1, exon 2, exon 3, exon 4, you're gonna stitch them together. Okay. So 1, 2, 3, 4, like that. Okay, next, you're gonna add a G cap on 5' end. Right? So a G cap. Okay? The G is the same as the guanine. And also, you're gonna add a poly A tail on 3' end. It's just a bunch of A's that you're gonna add. Okay, dot, 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 a bunch of A. The longer the tail, the longer how do I phrase this? The longer the tail, the longer it takes to break down the messenger RNA. Okay, maybe just stop copying for a second and listen to me explain. Remember, this mRNA is going to be needed to make proteins, right? Some proteins you are going to need to make for some time. Okay, so you want the RNA to last longer, so you give it a longer A tail. Okay, some proteins you're only going to need to make briefly. So those transcripts you're going to need just have short poly A tail. If it's really short, then it will break down very quickly. Then you won't be making the protein afterwards. Okay, so having different lengths for the poly A tail helps you have an extra layer of regulation, right? It will help you control how long to make the protein for. And that's it. Post-transcriptional modification. Just 3 small things you're gonna do. Okay? Remove the junk DNA, connect the exons. We are connect the exons, right? We are gonna also add the G prime cap on the 5' end to basically stabilize it, and then add a poly A tail to control how long it's gonna last in the cytoplasm. Okay, so that's pretty straightforward.
So Vincent, this happens in cytoplasm or
This happens in
inside?
the nucleus, yeah. Once we are finished, this is gonna exit the nucleus. Okay, and the finished product is the mRNA. This one here, that's the pre-mRNA. It's the premature mRNA. And then here you have the actual mRNA, okay?
Professor, excuse me, can we
Mhm.
add instead of AAA, for example, CGT?
Oh no, it's just the A. That's why it's called a poly A tail. It's a bunch of A's, yeah. Okay, let's go through some questions. Oh, this is just a picture. You can study the picture yourself, okay? But it's the same information. You see the little light pink stuff here? Those are the introns gonna be removed. You see the 5' cap and the poly A tail. And then it's gonna go to the cytoplasm. And then you're gonna have the ribosome to translate it. There's a bunch of other stuff that I have to tell you later. But we up to this point of the story, we are at the point where the RNA transcript is ready and it's going to exit into the cytoplasm. This is happening like all the time in the body, right? Anytime you need to make a protein, and you are making protein again, like all the time. Okay, can we quickly do these? Yeah, you know what, let's just do the poll. Let's see how. If no one's doing the poll, I'm just gonna skip that part. But let's see. Beginning of a gene. What is that? All right, most of you got it. It is the promoter. Very good. Next one. Okay, any last call? I have 6 people. I think it's the usual 6 people who are participating, which is it's all right. Yep, all of you got it right. It is the adenine. Okay, the 5' is the guanine, right? 3' is the A. What is removed during post-transcriptional modifications? Okay, correct answer is introns. Introns are insignificant. We remove them. Exons are expressed. We need them. All right. I am gonna skip the break today. If we have the break, we might not be able to finish the important stuff. Let's just push through for maybe another 25 minutes, 30 minutes, and then we'll end. Okay, I'm sorry for robbing your breaks. But that's the transcription process. Everything we learned so far. Now it's translation. Okay, so this is the ribosome and then all the stuff that are involved in the whole process. It's a mess. But let's review some of the key concepts that we know so far. I hope you know that nucleotides are the monomers of DNA and RNA. Okay. And each nucleotide is either ACTG or ACUG, depending on its DNA or RNA, right? So I'm sure you know that part. As for proteins, the monomers are amino acid, and there are 20 amino acids. 20 amino acids. So let's take a look at how we decode the DNA. We have the messenger RNA now. Okay. We have the messenger RNA now. And the messenger RNA is, of course, a copy of the coding strand. And so, for example, there's a template right here. It's just one of the genes. If the template is ACC, then the messenger RNA, hopefully you know now, is UGG. But what does UGG actually mean? Well, it turns out we read the nucleotides in triplets. Okay, so that's one triplet, another triplet, so on and so forth. And each triplet, every 3 nucleotide, is called a codon. And every codon, turns out, codes for a particular amino acid. So UGG over here actually codes for the amino acid TRP. It's tryptophan. Okay, they all have their short form. You don't have to know their full name. Okay, it's just easier for me to say the full name. That's why. So UGG codes for tryptophan, and then UUU codes for phenylalanine, and so on and so forth. So every 3 nucleotides, when grouped together, we have a codon. And the codon will tell us what amino acid to put together when we build the protein. So how many codons are there? Well, it turns out there are 64 codons. Let's take a look at how that's possible. We have 3 blanks, right? So how many possible letters can we put in the first blank? Well, it could be A, C, U, or G, right? So there are 4 combinations that you can put, 4 choices for the first blank. How many choices for the next blank? What can go into the next blank?
4.
That's right. Also A, C, U, G, right? And then another 4. So 4 times 4 is 16. 16 times 4 is 64. So there are a total of 64 codons. But there are only 20 amino acids. Okay, so meaning some amino acids are coded by more than 1 codon. So there's a little bit of redundancy here. And as you will see later on, probably not today, next week, that these redundancy helps protect us from mutation. Okay. Now, when it comes to translation, we also have to have a start and a stop. Okay. Transcription, we already know, right? Like promoter is start, terminator is stop. When it comes to translation, we also have to have a start and an end. So the start codon is AUG. You know, remember, like August, that's like the start of, you know, almost the start of the school year. Start codon, AUG. Okay. I want you to remember it because it will make it easier when you do certain questions. And then there are 3 stop codons, UGA, UAA, and UAG. So when the ribosome sees one of these stop codons, it will stop making the protein. Now, this is difficult to memorize, I suppose, but I have a cheesy way for you to remember this. Okay? You got A in biology. You are awesome. You are good. Okay? So you just remember these 3 sentences, and you have your stop codon. You got A. You are awesome. You are good. These are the stop codons. Okay? And trust me, memorizing the stop codon will be useful when we do mutations later on. Okay? Now, you're gonna have to have this sheet with you when you do the test. Okay, when you do the quizzes and whatnot. This is called the codon chart. Okay, codon chart. You want to print one out. You can just take it from the PowerPoint slide. Okay. And I'm gonna teach you how to use this codon chart. Okay. So this codon chart has all 64 codons. Okay, and then you know it has the amino acid that they correspond to. So let's take a look here. Right here is already the mRNA. Okay, it's already the mRNA. Let me just give myself some space here. Okay, it's already the, oh boy, I hate it when it does that. Okay, there we go. Yeah, I'm just gonna give myself some space is all. So let's see. The ribosome is gonna come, and then it sees AUG as the first codon. So this is how we look it up.
It said AUG is for all amino acids for the first
Okay,
codon.
yeah. Let me rephrase your question. I think you're asking me, is AUG the first amino acid? Is it the first codon for all the proteins? Is that?
Yes.
Yeah, it's correct. Yeah, because it's the start, right? It's the beginning, right? Okay. So we're gonna have to look up AUG, and this is how it works. Okay, let me see if this is gonna. Okay, so like first letter, right? We look at this row. Okay, so we find the first letter here. Okay, and because it's A, so that's the row that we're looking for. Second letter is U, right? So we look for the second letter up here, and U is right here. So I mean, you don't have to highlight it every time. I'm just showing you, right? So that's AU. So that means it is going to be somewhere in here. Okay, it's where the row and the column intersect, okay? And then to find the last one, we look at the G. So G is right here. Right? So we go all the way, and that's MET. Right? So the first amino acid is MET. Next is AUU. Let me do this one more time for you. Okay, so A, same row over here. U, same column. And then the last is U. So we just look at the U, and then we come all the way over here. So AUU codes for, I know this is difficult to read, but it's ILE. It's not LLE, okay? It's ILE. It's isoleucine. You just have to write the abbreviation, okay? Can someone tell me what CCU is? Okay, take your time, okay? Don't rush. Give other people a chance to look for it as well. What is CCU? Very good, Mary. Yep, Leah got it as well. Yep, Aminat. Right. So CC means it's here, right? And it's all pro anyways. But just for completion, CCU, right? That's proline. Okay, we're gonna go ahead and do another one. CA, which means it's in this block, and is U. Right, so CAU, that's histidine. Next one. No, actually, why don't you finish this? Why don't you keep going? Okay, very good. CGC. This is not too bad, right? I hope it certainly should be better than the template strand coding strand business. Huh? UUC is PHE, and then PHE And then UAA, you are awesome. That means it's a stop codon, so we stop. That's it. Okay. So this is what happens in the body. When the ribosome see these codons, it knows what amino acid we need to connect together. And then you're gonna build the protein. This protein is gonna have 1, 2, 3, 4, 5, 6, 7, 8 amino acids. Okay.
So Vincent, in this way, all the proteins start with MET.
That's right. Now I want to bring up another point. Like English, right? When you write a sentence in English, the first letter of the sentence must be capital, right? Does that mean you cannot have capital later on in the sentence?
No.
No, like you can have a name, you can have a place, you know, whatever, right? Like my English is not the best, so I don't know what you call those things, okay? But like if you have a name later on, you might have a capital letter in the middle of a sentence, huh? So same thing here. Even though we begin with MET, it doesn't necessarily mean you cannot have another MET later on within the protein. Okay, so don't be surprised if you see something like that. And also, in reality, the proteins are really long. Okay, so sometimes I might give you a sequence, and it might not necessarily be the entire protein. And some proteins are like thousands of amino acids long, right? Yeah, hundreds of it. So it's not possible to fit everything. So maybe I give you a short sequence that's in the middle. And in that case, it might not necessarily start with MET, and it might not necessarily start with the stop codon. So like, don't be surprised if that happens. Did you have a question, Leah? I saw you unmute.
Yes. Are we not gonna find the amino acid for the stop codon?
There is no amino acid. That's why it stops, right? When it gets to the stop codon, it literally has no amino acid corresponding to it. That's why the ribosome knows to stop. Like this is the end of the protein. Right? Okay, so like I said, right, like you have to have this chart, right, with you. Don't look it up during the quiz because you're gonna have like the lockdown browser, right? Like you might not have access to the internet necessarily, right? And don't have it on your phone. It's like so small you can't see. You need to, you know, print it out or something. Yeah, I know not everybody has a printer, but this is something that's worth printing out. Okay, go to the library or something. Okay. I just need to make sure everybody knows how to do this. So can I for once get like a full class participation on the poll? I want you to tell me what CGA codes for.
We need that table.
Oh, you don't have it? Okay, fine. Let me see what I can do. There we go. There we go. Okay, I'm missing 4 people. 3 now. Is everybody actually in class? Figure out sometimes people just log on and then I don't know where they go. Okay, I mean that's good. 75%. That's I think that's as high as I'll get. Any last one here? Last attempt? No. Okay. No. It's ARG. Great. Good job. Okay. Now this question is tricky. I don't want to do it now. Okay, it's gonna confuse you. We'll come back to it later. Okay. Remind me next class. I just want to do a few more examples here. Okay, this is example 1 is also very straightforward. It's just asking you to decode it. I know you can do it now because we just did the poll and everybody who participated got the right answer. So I'm certain you can look up these codes. Nothing special here. So I want to do 2 more examples with you, and then it will be bye-bye time. Here, example 2. Transcribe and translate the following template DNA. So now we gotta do it all the way from the start, right? All the way from the start. This is template. Excuse me, template. Now, this thing is, is not going away. It's coming back to bite us in the butt. So what is the mRNA? Do you have that sticky tag, the cheat sheet that I told you to make? Right? Or maybe you don't need it. But how do we go from template to messenger RNA? What's the relationship? Is it identical? Is it complementary? Which one is it, guys?
Complementary.
Complement.
That's right. So that's what we're going to do. T goes with A goes with U, and so on and so forth, right? Okay. For the interest of time, I'm just going to go through it, you know, relatively quickly here, but you should try this on your own later. And you have access to the recording, right? There you go. And then that's it. And then you can look up the amino acids. Now, here's the thing. You have to be careful on the test. You have to take your time to think about it. Because if your mRNA is wrong, then your amino acid is going to be wrong as well, right? So you have to make sure that when you convert from a DNA to RNA, you're actually doing it correctly. In any case, if you look up AUG, it's MET. CAU is histidine, right? GGA, UUA, so on and so forth. That's it. Any questions? Last one. Now we are given a coding strand. And that means we have to come up with the mRNA, don't we? And what's the word, guys?
Identical.
Yeah. Okay, now I feel a little bit better. At least I know some people get it. I would say a lot of you actually understand this much more than, you know, the last hour, right? So identical, that means we just copy it, but don't forget to change the T to U. Okay, and then you do it, right? Again, I think you are able to handle that. Now this one doesn't stop at the end. And like I said, don't freak out if it doesn't stop. It doesn't mean you're doing it wrong. It just means that, you know, it might be longer. In reality, it's much longer, often the case. Okay. So that's what I need you to kind of, you know, work on a little bit between now and next week. We also have the lab. The lab is pretty self-explanatory. Yeah, maybe I'll tell you a little bit about it. Okay, maybe I'll tell you a little bit about it since we have a bit of time. For next class though, I need you to print the study guide. Okay, you really need the study guide. I know a lot of you print it normally, but you know, if you normally don't, I need you to print it. If you have to pay for printing, then maybe print out this one here. Okay, you're gonna need this table, or you can copy it yourself. I don't know, whatever works for you. Okay, because we're gonna fill this in. We are also going to be doing these mutation questions, number 10, and also 11. Okay, so these are exercises that we're going to be doing in class next week. So that's why you have to have the sheet, right? Okay, I'm gonna go and talk to you about the lab for a little bit. and hopefully it will give you some clarity as to what is expected and so on and so forth. So here is the lab. If you go on eCentennial, as always, I'll post a link to it, of course, but content, virtual labs, and this is microscope. Okay, so there's a short video that you can watch and tell you how to use the microscope and how to take care of it. You can watch that on your own. There's a link to the virtual microscope, and then there is a lab. Okay, so you have to download the lab. And let me download the lab first, and we'll talk about it. So here it is. This is the lab. And you just follow it basically. Okay, it's a Word document. And I want you to type directly into the Word document. Don't change anything. Okay, you don't have to make it look nice. You don't have to title page or whatever. Just type directly into the document. Okay, like to make it easier for me, maybe type it in red or something. But it's okay. It's not a mandatory thing. Also, if you hand this in, you have to hand it in terms of a Word document. Any of you use Mac here, like Mac computers? Anybody at all? Apple stuff? Yeah, okay. So if you use Apple, you have to convert it into PDF before you send it to me. Or I think you can also export it in terms of as a PDF or as a Microsoft file. It's just that when you submit it using the Apple format, the dot page, I can't open it because you know, it's not compatible, right? So that's the only thing to watch out for. Okay. So it's pretty straightforward. It says launch the website. So you click on this. That's the virtual microscope. It's gonna give you a warning. Trust me, it's safe. So you just click onto it, and then it will take you to this virtual microscope. Now it's usually a little bit slow. I'm gonna turn off my video here to see if it's gonna be faster. It's a little bit slow when I'm showing you on Zoom. Okay, I don't know why. Okay, but it should be very fast when you are doing it on your own. Don't do it on your phone. You gotta do it on a computer. Okay, it's much more. It's much easier that way. And then you just follow the instruction. It says click on Learn. Okay, so you see there is a Learn button here. You click on it. Okay, and then you can click on these question marks. It will tell you, oh, this is called a course focus. And then the function is blah, 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 blah. You click on this, and it says this is the eyepiece, and the function is da, 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 da. So you just read them. And the first question here is asking you to match the names. Super easy. Okay. Which one is the arm? And you're like, oh, arm is, you find the corresponding letter. If you don't know what it is, you find it out here. Click on the question mark, you're like, oh, that's the on-off switch. That's not the arm. And then you just find it basically. You understand that? Okay. So there's the arm. And you're like, oh, okay, that's J. So you type in the letter J. Do you understand? Okay, I can't type. I don't know why for some reason.
Professor, for this lab, we just fill out this Word document and send you.
That's it. Yeah, you send this in, and then you sorry, you post it on the eCentennial. That's right. I'll give you the link as to where to post it. Okay.
You said you will give us the link for sending. I was just asking where should we send it to?
Yeah, have you guys handed in anything digitally in any course, like the Dropbox?
Yes.
Yes.
Yeah, it's like that. Okay, so I'll create a Dropbox and you put it in. Okay, that's all.
Yeah.
Very easy. Don't email it to me. Okay, if you email it to me, they get lost in the shuffle. It's like, yeah, just submit it properly. Okay, and then part 2, right? And then you go to the main menu. Okay, click on export. You just follow it. It's very easy. Right? So I'm going to show you. Click main. Okay, explore. All the instruction is in the file. Okay, you open the box and you start with sample slide, letter E. Okay, so it's a little bit blurry because like in a real microscope, you have to focus it, right? So usually you will focus it with the course first. And you can see over here, the stage is actually moving. Right? That's what the course knob does. It moves the stage up and down. Okay, and then it comes into focus, and then you can adjust it a little bit. You can even turn up the light or turn lower the light to adjust for the brightness. I know it's not the real thing, and it seems silly to play around with it like on a computer, but that's the best we can do. Okay, actually, believe me, I am trying very hard to create another lab activity, but it's not that easy. Okay? It has to be something that relates to what we're doing, and it cannot be too difficult, right? So I'm working on that. If you have any suggestions, right, please feel free to email me. Right? I would love to hear what you think from a student's perspective. But for now, this is what we are stuck with. Okay, and so it says here, take a screenshot for the E at 4 times magnification. Okay. So a couple ways you can do it. You can take it with your phone and then paste it into the document. If you use PC, there's actually something called a snipping tool. You just type in snipping, okay, and then you launch it. Then you can copy it. There, you copy it, right? And you can Control C to copy it, Control C, and then you go back to here and then you paste it. Now, of course, it gets really big, right? So don't forget to resize it. That's all I'm looking for. Okay, don't rotate it because this is what it actually looks like under the microscope. And then you do it for 10 times and 40 times, right? So look here. You can go back to here and you can go up the magnification. Okay? Simple? I don't know what the screenshot button is on Apple, but I'm sure you will figure it out. Okay. Simple Google search would be all right. Okay, here it's asking you what is the relationship between the image and the object. Okay, so what do you think? Like how does the normal E, that's the object, and this one is the image. What's the relationship? Tell me. We'll do this together. What's the relationship?
It's inverse.
Okay. I think you might be looking for another word there, but I kind of get what you mean. Anyone else? Like what do you mean by that? Like, can you give me some more description? What do you mean by inverse?
Not like it's upside down.
It's upside. Okay, yeah. So the image is actually called inverted, meaning it's upside down and it's backward. And not just upside down, it's also backward, right? Another way to think of it is that it has been rotated 180 degrees. Okay. You can use that if you want. You can clean it up a little bit, but that's the idea, right? So here it says, besides the letter E, give 2 other letters that would be appropriate for this activity. Okay, so I'll give you an example of something that's not appropriate. Okay, if you use the letter S, that's not appropriate. What does it mean by not appropriate? It means like if you put the S under the microscope and you look at it, it will still look like the original. Because the letter S, when you rotate 180 degrees, it looks exactly the same as the original, doesn't it? Do you understand?
Yeah.
Yeah, so that's not a good letter to use. Okay. All right, so like you tell me, 2 other letters that's appropriate. All right, that's it. And then there's some calculations here. Just read the stuff, it will make sense. Okay, you should be able to figure this out. And then there are some calculations going on here. Okay. You know what? I don't have time to go through it right now. But I think I will explain this more on Monday. When do I see you guys? Monday, yeah. I'll explain this more on Monday. Okay, but that's what the microscope lab is. Okay, so you can start on it this weekend. Do as much as you can. I will go through the calculations with you on Monday. And that would, help you clean it up a little bit. Any questions?
Professor, the due date for the quiz and lab. when
Yeah,
is-
yeah. So the lab is gonna be due. You know what? I can move it to Friday, no problem. Okay, I'll say the lab is due at the end of Friday. And in terms of the quiz, I think let me see. I'm gonna finish teaching 7 on Monday. I think it's okay to put the quiz on Wednesday. Okay, I'll put the quiz on Wednesday, but I'll open it until Thursday. How about that? Okay, so if you want to do it on Wednesday, you can. But then if you want to wait and do it on Thursday, that's okay as well. Does that sound fair?
Yes.
Yes.
Yeah.
Yes.
Okay. Yeah. So lab's gonna be due Friday. I'll explain how to do the calculations on Monday, but you should definitely start on it today. Quiz available Wednesday to Thursday. No class on Wednesday then. Okay, no class on Wednesday. Wednesday you can just spend it working on your lab and just preparing for the quiz. Okay, but Monday, please come to class on Monday. Okay, no class. I'll post all these changes on eCentino afterwards. Okay, that's it. Thank you very much for coming. You guys are always, good with attendance, this particular group. So I thank you for that. Enjoy the rest of your week.
Thank you.
Yeah.
Thank
Bye-bye.
you.
Professor. Yeah.